I am going to talk about two mRNA decay pathways today, and this derives largely from work that's done in my lab. The first one is called Nonsense-Mediated mRNA decay, which we abbreviate NMD. This is a translation-dependent quality control pathway. So you've already heard about proofreading of a number of molecules during splicing, for example, and translation in the sense of the ribosome. And I'm going to be talking about a different type of proofreading mechanism called NMD. I just want to review that translation normally terminates at one of three nonsense codons, which are shown here in red. These codons usually do not encode any amino acid, and they signal the release of the nascent protein and ultimately dissociation of the two ribosomal subunits from the mRNA template. We began studying NMD many years ago, and we made two important findings. The first was by studying human diseases that we found that many diseases are due to frame shifter nonsense mutations that result in the premature termination of translation. And the disease is due because there is an absence of full-length functional protein. We also found that the cell has a way to eliminate mRNAs that prematurely terminate translation, and that is the process of NMD. And we think this exists because those mRNAs that prematurely terminate translation can generate a truncated protein, and truncated proteins can be toxic to cells. You'll recognize this to be a very simple overview of the steps involved in mammalian gene expression for genes that encode proteins. And these steps of RNA metabolism are often linked mechanistically, temporally and mechanistically. And I want to provide some examples of that. In our studies of NMD, we found links between pre-mRNA splicing in the nucleus and mRNA translation and degradation in the cytoplasm. Work done by Hervé Valliere when he was a postdoc in the lab. We were working at the time with Melissa Moore. Found that the links are provided by exon junction complexes, or EJCs, which are complexes of proteins deposited upstream of the exon-exon junctions of newly synthesized spliced mRNAs. And the EJCs stably contain some of the NMD factors that I'm going to be talking about. Yasuo Ishigaki, when he was a postdoc in the lab, discovered a new template for protein synthesis that we call the Pioneer Translation Initiation Complex. You'll see that NMD in mammalian cells is a consequence of nonsense codon recognition, of premature termination, PTC codon recognition, during a pioneer round of translation. This initiation complex contains the cap-binding protein heterodimer CBP80 and CBP20. Notice it's different than EIF4E, or what Marv called 4E, which is the cap-binding protein on the bulk of cellular mRNA. This is a cap-binding complex that is particular to newly synthesized mRNA, and you'll note that it is acquired by pre-mRNA. This pioneer initiation complex not only contains the cap-binding protein heterodimer CBP80 and CBP20, but if the mRNA underwent splicing, it will contain the post-splicing EJCs. Even though we uncovered NMD by studying human disease, we think it largely functions to eliminate transcripts that are made by mistake all the time. And that is because the resulting truncated proteins, while in a number of cases are unstable, they can be stable in certain circumstances and function in dominant negative ways or other deleterious ways to cells. And so NMD can be viewed as a type of quality control or mRNA surveillance mechanism. And it makes sense for cells to survey their mRNAs very early in mRNA biogenesis and eliminate those that have the potential to encode a toxic truncated protein, hence NMD, targeting the pioneer round of translation. Here's an example of what happens that's bad when NMD fails to occur. And the example pertains to the human beta-globin gene. The gene consists of 3 exons. It initiates translation, of course, in the RNA in exon 1 and terminates translation normally in exon 3. Nonsense codons in the final exon of the beta-globin gene, even though they are premature termination codons or PTCs, do not trigger NMD. And as a result, the consequence is a dominantly inherited form of what is usually a recessive thalassemia disease. You're going to see in a minute why PTCs in the last exon do not trigger NMD. The mRNA is stable. It turns out the truncated beta-globin protein is stable. But this truncated beta-globin protein functions very differently than the full-length beta-globin protein, which together with alpha-globin makes up hemoglobin. The truncated beta-globin protein can only dimerize with alpha-globin. And this results in ineffective erythropoiesis and a dominantly inherited form of what is usually a recessive disease. All right, so you can imagine we were very interested in understanding how cells know whether a termination codon should trigger NMD or not. And so we moved premature termination codons around within genes, and we moved introns around and established the 50 to 55 nucleotide rule. What this rule says is if there is a PTC in the purple region here, then there generally is NMD. That means the PTC will be situated more than 50 to 55 nucleotides upstream of the last exon-exon junction. We don't think there's anything special about this junction. We think all junctions will trigger NMD if there's a PTC this distance upstream. It's just that if you're looking at your favorite mRNA and you have a PTC and you're asking, well, will it trigger NMD or not, you would look 50 to 55 nucleotides upstream of the last exon-exon junction. And then PTCs in the light blue region do not trigger NMD generally. So you can see why PTCs in the last exon of the beta-globin gene do not trigger NMD, according to this rule. This rule predicts that mRNAs that have a PTC but did not undergo splicing, let's say they derive from an intron-less gene, they'll be immune to NMD, and that is true. All right, I just want to review, since my time is very limited, the idea that NMD protects cells from mRNAs that prematurely terminate translation. So even when the disease results from an NMD-induced protein deficiency, there's not enough full-length functional protein, the disease phenotype may be milder than and different from that caused by an expressed truncated protein. So NMD protects many heterozygous carriers of PTC-containing genes from manifesting disease phenotypes that would result if that truncated protein were produced. So we've already talked about what happens when PTCs exist 5' to the NMD boundary, that 50 to 55 nucleotide position upstream of the last exon-exon junction. The recessively inherited mutations in the case of beta-globin, but 3' to the boundary, there's a dominantly inherited form of the disease. And that's true for many diseases, many more than are that are on this list. And that's because I think you all know that 2 different individuals can manifest the same disease phenotype, but the molecular basis of that disease, the actual mutations within the affected gene, can be different. All right, it turns out that most mRNAs targeted for NMD derive from alternative splicing. About 1/3 of all alternatively spliced human transcripts are targeted for NMD, which is remarkable when you think that conservatively 90% of human genes encode pre-mRNAs that undergo alternative splicing. And here's one of a number of scenarios by which NMD can occur by alternative splicing. In this example, the alternative exon is red. Normally, it's removed by splicing, so the translation initiates and terminates normally. There's no NMD. But if that red exon were retained and it had a stop codon, a PTC in frame with the initiation codon, then there would most likely be NMD. or if the intron were retained and it didn't consist of a multiple of 3 nucleotides, so there was a shift in the reading frame, there would most likely be the generation of a PTC downstream of the shift. And if there were an exon-exon junction sufficiently downstream, NMD. So this type of alternative splicing can be viewed as a type of intron retention, and other types of alternative splicing that can lead to NMD would be exon skipping. So let's say that's not a good example, but let's say the red exon were skipped and it's normally supposed to be there, but it's not a multiple of 3 nucleotides. That results in a shift in the reading frame. And also if there's splicing downstream of the normal termination codon, there would be NMD. So it's less important as to whether the termination codon is premature or normal, and more important as to whether it is followed more than 50 to 55 nucleotides downstream by an intron, and therefore, after splicing, an exon-exon junction. Are you following this? I'm talking faster than normal.
Is it okay?
All right.
Do you have a question now?
Yeah, do you have questions? Yes.
The last slide again.
Yes.
About the NMD, is it like controlling the expression?
Yes. So NMD will control the level of gene expression in the sense that it will down-regulate the amount of mRNA that is produced. The protein will always be truncated if the mRNA contains a PTC. And I should also tell you that NMD is not 100% efficient. So it reduces mRNAs to maybe 5 to 10% of their normal abundance. About 5 to 10% of NMD targets escape NMD and get remodeled and do produce truncated protein, but it's a low level now. Okay, from work that we did with Ben Blenko, it turns out that most of the NMD targets generated by alternative splicing are mistakes, they're junk. And we know this because of a number of reasons, but I'm just listing 2 here, and one is that their level is so low that they're not likely to contribute to physiological cellular processes. And also there's rarely tissue-specific regulation. And you know, all of you who study alternative splicing or splicing, that alternative splicing expands our proteome in the sense that we have only 20,000 genes. And we need an expanded proteome to have our developmental and organismal complexity. All right. I just want to mention that NMD can be used constructively. So here's a nice example of your question to regulate gene expression in a pathway that's called regulated unproductive splicing in translation, or alternative splicing coupled to NMD. And this type of regulation typifies a number of splicing factors, including the serine arginine-rich splicing factors and some H and RNP proteins, indicating there are important feedback mechanisms. And the example I'll give is for poly-permitting track binding protein. So this is a splicing factor. It is encoded normally by an mRNA that retains exon 11. But when intracellular levels of PTB become too high, PTB feeds back on the splicing of its own pre-mRNA, resulting in the skipping of exon 11, a shift in the reading frame, because exon 11 is in the coding region, and the generation of a PTC in exon 12. It's certainly not the last exon, and therefore there's NMD. And then there are natural NMD targets. NMD will trigger physiologic mRNAs that have an open upstream reading frame and upstream open reading frame. We've already talked about mRNAs that naturally contain an intron downstream of the normal termination codon. And we've studied selenoprotein mRNAs where the UGA codon in these specialized mRNAs is not a termination codon all of the time. Depending upon intracellular selenium concentrations, there can be recognition of the UGA codon as a selenocysteine codon. And that's because there's a selenocysteine insertion element in the 3' translator region of these specialized mRNAs. All right, here we start with the mechanism. We've already said that NMD occurs during a pioneer round of translation. There's the PTC. This is going to trigger NMD if it resides at least 50 to 55 nucleotides upstream of an exon-exon junction that has an EJC upstream. And Hervé positioned the EJC about 20 to 24 nucleotides upstream of the junction. When you think about it, the 50 to 55 nucleotide rule makes sense, because if a ribosome got any closer than 50 to 55 nucleotides to this junction, its leading edge will physically remove the EJC. And you have to have the EJC there for the choreography of events I'm going to tell you about. All right, so this PTC will trigger NMD. It associates with a complex called SURF. The two translation termination factors are the ERF, the RF. UPF1 is a helicase, very important for NMD. And SMUG1 is a kinase that phosphorylates UPF1 at the appropriate time. So SURF, that was initially described by a Japanese group in Yokohama. And then UPF1 and its kinase move to the EJC, at which point SMUG1 phosphorylates UPF1. There's the step of translational repression and decay. So I want to talk about translational repression. This was done by Olaf Isken when he was a postdoc in the lab. He's now in Germany with his own lab. So I've gotten to the point where UPF1 is phosphorylated by its kinase, SMUG1. They're both on the EJC. And then, interestingly, phospho-UPF1 interacts with the two highest molecular weight subunits of this initiation factor. So Marv showed you this kind of stuff this morning. This is a 43S pre-initiation complex poised at the initiation codon, waiting to have the 60S ribosomal subunit join so that translation can initiate. But that doesn't happen because phospho-UPF1 prevents 60S ribosomal subunit joining. No functional ADS ribosome is formed, so there's an inhibition of translation initiation, further initiation during this pioneer round of translation, and also phospho-UPF1 recruits the degradative activities that we talked about yesterday. And this is a remarkable translational repression step. To prove it further, for example, Olaf showed that a PTC-containing mRNA that initiates translation using an internal ribosome entry site that doesn't use EIF3 is immune to NMD. Whereas if you put another translation initiation process that depends upon EIF3 into that exact same message, you get NMD. So translational repression is very important for decay. And that's often you'll see that with microRNA-mediated decay and other decay processes. Translational repression is important. All right, so I've been telling you that NMD targets this mRNP, which Fabrice Lejeune, a postdoc in the lab, showed is a precursor to, this is the steady state RNA. Remember, it's got EIF4E, or what Marv called 4E at the cap. And when you think about it, it makes sense that NMD would target this mRNP. And by the time the RNA is remodeled to this, it's immune to NMD. So it's immune because there's no EJCs and associated NMD factors, which are UPF factors. They're gone. And also, CBP80 actively promotes NMD. And that is work that was done by Zhongguo Kwang when he was in the lab. He showed that UPF1, the helicase, forms an association with CBP80, the cap-binding protein, part of that heterodimer that typifies newly synthesized mRNA, and promotes NMD. And so I'm going to show you a little bit more of a involved mechanism. Here's a transient or weak interaction of the helicase with CBP80. And during that pioneer round of translation, the SURF complex formation is activated by the ability of CBP80 to interact with UPF1. CBP80 actually escorts UPF1 and its kinase to the two termination factors to form SURF and CBP80 associated with it in an RNase-insensitive complex. And then CBP80 escorts UPF1 and SMUG1 to the EJC, which results in UPF1 phosphorylation, repression, and decay. So if I'm limited in time, maybe you don't want to know how we did these experiments because they're published. But let me just tell you what Jung-Wook did. If you want to figure out the importance of a protein-protein interaction in a mechanism, what you can do is map the region on each protein that's required for the interaction and then express in cells one of those regions. And you want the region to be as small as possible because you don't want it to mess up any other cellular process or any other proteins that are interacting with the protein whose little interface for the interaction you're interested in inhibiting. I've said that too long, so God knows what I wanted to say next. But anyway, so what Jung-Wook did was he mapped the region of CBP80 that interacts with UPF1. He also mapped the region of UPF1 that interacts with CBP80, and he expressed each as a little piece separately and could inhibit this interaction, and he found that he inhibited the CBP80 escorting to form SURF and CBP80 escorting to form SMUG1 and UPF1 at the EJC. Okay? And as I said, this is published in Molecular Cells, so you can look at it, and if you have any questions, you can send me an email. And that saves us a lot of time. Okay. I just want to mention that the restriction of NMD to newly synthesized mRNA appears to be unique to mammalian cells. We've done work with Fred Sherman showing that NMD in yeast, Saccharomyces cerevisiae, targets both newly synthesized and steady-state RNA. And because this was controversial, and the Pioneer round was controversial, and intron position influencing which termination codons trigger decay of mRNA was controversial, we always felt that we had to really be sure we were correct. And one way we felt confident about our conclusions was to tether UPF1 downstream of a normal termination codon and mimic the last step of the EJC, right? What the EJC does last is it recruits UPF1. And when we do that, we bypass the need for CBP80, which is involved in escorting UPF1, and also the EJC. So by so doing, we can target both MR and Ps for NMD. Okay? Okay, so now, how am I doing?
You have, see, another, I tell you, 5 minutes.
I have 5 minutes left?
No, you have 10, but behave like if you have 5.
Okay. So the second pathway that I want to talk about was discovered by Yoon-Ki Kim when he was in the lab, and it involves Stauffen. Marv talked about Stauffen in oocytes, and I'm talking about Stauffen-1, which is a paralog to the Stauffen that, it's an ortholog to the Stauffen that Marv talked about in mammalian cells. And it, when it binds to an mRNA 3' UTR, will trigger mRNA decay. And I'll tell you how. But you may say, well, how the heck did he figure that out? And what he did was he used yeast 2 hybrid. Marv talked about yeast 3 hybrid. I'm talking about the yeast 2 hybrid analysis that uses yeast to look, in our case, at interactions of mammalian UPF1, human UPF1, with human proteins. And what Yoon-Ki came up with was Stauffen-1. And we were hoping to get a bunch of factors that were involved in NMD, but instead discovered a related pathway, Stauffen-1 mediated mRNA decay. So in SMD, Stauffen binding to an mRNA 3' untranslated region functions analogously to how the EJC functions during NMD, in the sense that when translation terminates sufficiently upstream of the Stauffen binding site so that Stauffen is not removed by the terminating ribosome, then it will trigger mRNA decay because it can interact directly with UPF1. It can recruit UPF1 directly. And so this pathway, therefore, does not require EJCs. It doesn't require splicing. It'll target mRNAs that derive from an intronless gene. It doesn't require UPF2 or UPF3 or 3X that are NMD factors that are stable constituents of the EJC and required for NMD. And it targets both newly synthesized and steady-state RNA. Remember, mRNA bound at the cap by EIF4E, or what Marv called 4E, constitutes the bulk of cellular mRNA. And we think this is a pathway that's used by cells to conditionally regulate the expression of genes having a Stauffen-1 binding site in the 3' UTR of the product mRNA. And as we talked about yesterday in terms of general themes of mRNA decay, if you want to eliminate gene expression, you do it transcriptionally as well as post-transcriptionally through mRNA decay, and you have to target both mRNPs, certainly the one that constitutes the bulk of cellular mRNA. You have to get rid of it so it stops producing protein. All right, so once we realize that Stauffen binding to an mRNA 3' UTR triggered mRNA decay, we realized that the best way to identify SMD targets was to look at those that are upregulated when Stauffen was downregulated using small interfering RNA, siRNA, which is a technique we use a lot. And we did this work in collaboration with Luc Desgrossier at Montreal and found that in 3 independent microarray analyses where Stauffen was downregulated, conservatively 1.6% of HeLa cell transcripts, human cell transcripts, were upregulated at least 1.8-fold. All of these may not be direct targets of SMD, but many certainly are. And here's a short list of direct targets. So we're going to talk about Serpine-1 mRNA. This is involved in wound healing. It encodes a protein that's involved in wound healing. Interleukin-7 receptor mRNA is a target. And we've already talked about c-Jun, which is a proto-oncogene protein. And one of the last things Junke did before he left the lab was he showed that during the differentiation of C2C12 mouse myoblasts to multinucleated myotubes, the efficiency of SMD goes up. So I just want to tell you 2 stories now, and they're both done by a very talented graduate student in the lab, Chengguang Gong, who we call Gong, because I don't think we say Chengguang anywhere near close to the way it's supposed to be said. So it turns out SMD and NMD are competitive pathways in a way that is important for myogenesis. This is how they're competitive pathways. There's a CH-rich domain of UPF1 that can interact either with UPF2 during NMD. UPF2, remember, is a stable constituent of the EJC, important for NMD, or with Stauffen, which is important for SMD. And because of the either-or interaction, the 2 pathways are competitive. UPF2 and Stauffen compete for binding with UPF1. And what Gong found is that competition explains how the efficiency of SMD is upregulated during myogenesis. It turns out that during myogenesis, the abundance of STAW1 actually decreases. So if you just had that information in a vacuum, you would say the efficiency of SMD decreases during myogenesis, right? But it turns out the abundance of the competing protein, UPF2, decreases even more. So SMD wins out. The efficiency of SMD goes up during myogenesis. And this is important because during the differentiation of myoblasts to myotubes, PAX3 mRNA, Gong found is an SMD target. And it's important to get rid of PAX3 protein during this differentiation process because PAX3 protein maintains myoblasts in an undifferentiated state. Sara, yeah?
What's meant by myogenesis?
Myogenesis is, that's a good question, muscle cell differentiation. So mononucleated myoblasts are a precursor cell, a more primitive cell, an undifferentiated precursor to multinucleated myotubes. And because I don't have time to show you data, I'm not. Glauco's not protesting. And then during the process of...
You are my favorite, you know, but I have a control.
I know. So during the differentiation process, the efficiency of NMD goes down, and this is important because myogenin mRNA, which is critical for the myogenic process, is an NMD target. And it's one of these natural NMD targets. The mRNA has an upstream open reading frame, upstream of the open reading frame that encodes myogenin per se. So I think you're seeing how this competition between two pathways that have a common factor, UPF1, is really important for the myogenic process. It goes way beyond this. I'm just giving you a taste. And then I'm getting to my final story, which I think is a pretty exciting story, that talks about long non-coding RNAs. So, you know, almost all of the human genome is transcribed into RNA, and we don't know what most of the RNA does. And an example of a category of RNAs whose function we don't really understand is the long non-coding RNA category. And what Gong showed here is that long non-coding RNAs can actually transactivate SMD by duplexing with mRNA 3' UTRs via ALU elements in the ALU mRNA that are long non-coding RNA that are complementary to the ALU element in the mRNA 3' UTR. All right, so here was our dilemma. This is the only known SBS for all of our SMD targets. It's a 19 base pair stem with a 100 nucleotide apex. We knew this in 2007. We knew that if you disrupted the duplex, you disrupted SMD. And if you restored the duplex, but with completely different nucleotides, you restored SMD pretty well. The apex was somehow important. When that's deleted, we lost Stauffen binding to about 50% of normal. And Stauffen will bind to double-stranded RNA. Did I tell you it's a double-stranded RNA binding protein? It's a double-stranded RNA binding protein. So it will bind to double-stranded RNA. If you put double-stranded RNA in the test tube, it's going to bind it. So to look for specific binding, we need to do the binding studies in cells. We have to introduce Stauffen into cells, do an immunoprecipitation, so we precipitate Stauffen, and then look at the associated RNAs. And to be really sure, we cross-link before we lyse the cells so that any protein-RNA interactions that we see were formed in the cell and not as an artifactual association after cell lysis. But we couldn't find any structure that looked like that 19 base pair stem. And so what Gong noticed is that SMD targets, at least two, in fact, he's a computational biologist, it turns out, and he found that SMD targets are enriched in ALU elements in their 3' UTRs. So remember, Serpine 1 mRNA encodes a wound healing protein. No one knows the function of the protein encoded by this mRNA, but the red is the ALU element in the 3' UTR of each. And Gong found that of the putative SMD targets, those mRNAs that are upregulated in cells when Stauffen is downregulated, 13% contain a single 3' UTR ALU element. But if he looked at the bulk of cellular mRNAs by looking at a database, he found only 4% have one or more 3' UTR ALU elements. So remember we talked about yesterday how conservation in sequence between species or in a class of RNAs indicates function. So we thought this was an important finding. And so Gong hypothesized, let me tell you what ALU elements are. These are short interspersed elements or signs. They're particular to the primate genome. So they're not in mouse or rodents. They're about 300 base pairs, 1.4 million copies per human genome, very abundant. They're commonly found in introns and intergenic genomic regions, but I'm talking about ALU elements in mRNA 3' UTRs. And they regulate gene expression at multiple levels. They can create polyadylation sites or splice sites. I'm talking about creating Stauffen sites, Stauffen binding sites. So Gong hypothesized that some Stauffen binding sites derived from the intermolecular base pairing between an mRNA 3' UTR ALU element and a partially complementary ALU element in another transcript. He thought that other transcript could be long non-coding RNA, And he found that 378 long non-coding RNAs in Leonard Lipovich's published pipeline contained a single ALU element. So sure enough, by computational biology, he found this long non-coding RNA was able to anneal with pretty decent stability to the ALU element in serpine-1 and FLJ mRNAs. So the question is, did this happen in cells? And if it did, what was the consequence to the mRNA metabolism of serpine-1 and FLJ mRNA? So first he had to express the long non-coding RNA so he could manipulate it. And to cut to the quick, he used RNase mapping. He found there was a short and a long isoform that differed only in their 5' end. The short isoform was the more abundant. It's largely cytoplasmic and polyadenylated, excellent at cytoplasmic for a number of reasons, one of which is we're studying a decay pathway that occurs in the cytoplasm. It's dependent upon translation. We don't think there's translation in the nucleus. And here is a picture, a sequence of the short non-coding RNA, long non-coding RNA. There's the ALU element in pink. It's polyadenylated, and there's the AAA polyadenylation signal. And the model is in the cytoplasm, there are mRNAs, and there are long non-coding RNAs that can form a Stauffen binding site, each contributing a half of the Stauffen binding site. So we call the long non-coding RNA a half SBS RNA. And that duplex binds Stauffen. Stauffen actually stabilizes the duplex. And when translation terminates normally, because Stauffen can recruit UPF1, there's SMD. So this is a new function for long non-coding RNAs and a new function for ALU elements. And let me just show you, you're not going like this yet. What does that mean, Glauco?
Well, you are already 10 minutes late.
I am?
No, you are finished because...
I'm finished.
Yeah, because the next talk is...
I'm finished.
5:15.
Okay, I'm finished.
No,
But you can read it. It's published.
No, but, okay, we give you more minutes. How many do you need?
No, so let me just say, I'll just say that we were able to purify the complex, and you can read it. So this happens. I wouldn't be telling you if it didn't. We can purify the complex, and yes, these RNAs are in it. Stauffen, UPF1 is in it. Non-specific RNAs and proteins, not in it. And it's not particular to that long non-coding RNA, and it's not particular to those mRNAs. But I want to show you something cool. So this is a wound healing assay, and then I'm done. This is a wound healing assay where human keratinocytes are put in culture at 0 hour, then they're scratched. So you have a confluent series of cells, they're scratched. And these cells were treated with control siRNA, that half SBS RNA that I've talked about, serpine-1, which is involved in wound healing, it's downregulated, and this other mRNA is downregulated, it's involved in wound healing. And look after 16 hours, here's the normal wound healing rate. There's just a little gap. Look at the giant gaps here. And look, downregulating the half SBS made the wound heal faster. So these things do function. And I'm done. I'm just going to show you the guys again, and thank you.
Thank you.